Well, namaskar, good morning, and Shubham Nebu Gade Vandana. First of all, I would like to thank the organizers, and particularly Dr. Rao, for the opportunity to share our thoughts and findings in the area of inflammation induced chemoresistance in cancer cells. But this morning, due to time restrictions, it's already 4 minutes. I will focus mainly on the topic of chemoresistance, as that has been an area of research in my lab for several years. We have been trying to develop the models to understand the principles of drug resistance in cancer cells, mainly in the lab. And recently, we are trying to apply those principles in the clinical oncology. There have been a lot of challenges, but hopefully it will be a rewarding effort. Let me start with a kind of 30,000 feet view of the challenges which exist in translational cancer research. The first and most important is to identify causes of cancer and to intervene or intercede these causes to prevent the initiation of cancer development. And we have, I think this issue has been discussed. In India, this is particularly very important because by understanding the factors which are really causing the cancer in the local population and to prevent and educate the public, I think that itself will take care of 70% of cancer in this country. Number two is to enumerate and understand the molecular alterations that can be used as targets for cancer-specific therapy and use these to improve the treatment of cancer. And as you understand, there's a lot of emphasis, there's a lot of effort, there's a lot of money which is being spent in this area. And there are actually targeted therapies which have been developed. But I think this is mainly focusing on treating the early stage cancers when the cancer cell is really rapidly dividing. So third one is to discover why some cancers fail to respond to therapy and or relapse after treatment and to find the ways to improve responses. I think this is another very important area because more than 90% of cancer-associated deaths are related to metastasis. And unfortunately, only 1% of effort is spent to understand the process of metastasis. Virtually all the cancer deaths can be said to have occurred because the therapy failed, because these patients, they did not respond to the existing arsenals of anti-cancer, so they died. And particularly in India, I think it's even more important to understand the principles of drug resistance and metastasis because about 70% of patients, they die within first year of diagnosis because by the time patient comes to see the clinical or clinician, it's already too late. The disease has metastasized and has become refractory to existing therapies. So the issue of drug resistance is very complex. As you can see, there are about 360 genes which have been linked with drug resistance in cancer cells. And each of these genes, basically they promote drug resistance to a specific type of drug. And drug resistance can be induced either by change in the hundreds of molecules which are involved in uptake of drugs and to carry them inside the cell. Or on the other hand, it can be induced by overexpression of so-called efflux proteins like PGP, MRP. And there are about 48 members in this family which belongs to efflux pumps. And in between, anything can promote the drug resistance, including the drug inactivation, altered drug metabolism, and then change in the target against which a particular therapy has been directed. So that can also lead to the resistance against a specific target therapy, and of course, all these apoptosis and autophagy can contribute to the drug resistance. We put together a monographs on the issue of drug resistance in cancer cells, in which we covered actually several of the topics related to the drug resistance, including like metastasis and drug resistance. So if you have time, please read over this. So when the patient comes to the clinic with a refractory tumor, the clinician always sees that patient or that issue as a solid arrow pointing to in one direction. That means the clinician think that there's nothing going to work for this patient, and as a result, they start using very aggressive treatment protocols. Now, those protocols are not going to help patients. On the contrary, it's going to damage, it's going to cause more damage than the help. But I'm here to really discuss what we feel that it's not really a solid line pointing to one direction. It's a reversible process. And why I say that? There is an anecdotal evidence. I'll come to this later. Where we know that so-called the drug holidays, if you stop treatment for some time, and now you go back and treat the patient, these tumors, they start responding to some extent. Number two, response to epigenetic agents. There are some clinical trials where the epigenetic agents to activate those hypermethylated genes have shown some promise. And even the cell line models, we know that when you raise a resistance to a cell line, you always maintain that cell line under continuous pressure of drug. If you culture these cells for a long time without drug, they revert back. So that clearly shows that drug resistance is a reversible process. And let me give you some kind of analogy. The initial mutations in the cancer cells is like a hardware of a computer. So once that change occurs, nothing is going to change that. So it's a permanent change. But in order to adjust to the changing environments, these cancer cells, they use the epigenetic alterations. It's like a software of a computer. And software can always be hacked, it can be modified, and it can be corrupted. And that's exactly what tumor cells are doing. By using epigenetic alterations, they are corrupting the system. But at the same time, we know that software can be fixed. And the same thing, this drug resistance can be reversed and fixed. So, but in order to really understand this, we need to identify the drug-resistant epigenetically modified genes using biopsy samples. And multiplicity would be better in this case than the specificity, in the sense that the targets or with the genes which you identify should simultaneously inhibit multiple pharmacological targets which are crucial for cellular metabolism and energy status. I think if we really understand those genes, we have a very good shot at reversing the process of chemoresistance. So with that in mind, we started with a very simple model. We got a cell line, which is a transformed memory cancer cell line, and we, in the lab, made it resistant by weaning it through increasing concentration of drug, so-called adriamycin. And eventually, we picked a subline, which is now about 70 or 150 times more resistant than the parental cell line where we started. And we use this as a model to understand the genomic and proteomic analysis and came out with one gene which really stood out. It's a pro-inflammatory stress response gene called transglutaminase 2, or in short, it's called TG2. So you can see that when we did verified or validated this gene, it turned out to be that drug-sensitive cell lines, they totally lack the expression, but drug-resistant cells, they have a very high expression of this protein. And then we went to the NCI60 cell line model to see how this expression of this gene correlates. And we were really amazed that very specifically, irrespective of the type of cancer, the expression of TG2 was associated or was seen in those cell lines which were either highly aggressive, highly metastatic, or resistant to chemotherapy. So far, so good. So we were really excited with this information from public domain. Even more interesting, wherever you see the high expression of TG2, there's a cluster of genes which are upregulated. And there's a cluster of genes which is downregulated. As you can see, the reverse, those which have low TG2 expression, these genes are downregulated, and these genes are upregulated. So that gave us an idea that it might be the protein which is controlling multiple pathways, and that's exactly what we wanted. We studied other cell lines and patient tumors, and once again, very selectively, those cell lines which are either highly metastatic or resistant to chemotherapy, they selectively showed the high expression of this protein. In patients, these are primary tumors from breast cancer, and these are metastatic tumors. You can see clearly substantial increase in the expression of this protein. These are ovarian cancers, once again, resistant and invasive tumor cell lines. They have high expression. And this is low-grade tumor. This is high-grade tumor. And pancreatic cancer cell lines, which exhibit inherent resistance and aggressiveness, about 80% of these cell lines had a very high expression, basal expression of TG2 protein. And this is a patient sample. This is a normal duct, and this is a pancreatic ductal adenocarcinoma. More importantly, I'm sorry for this slide, it's a little kind of fade. More importantly, the expression of TG2 in tumor cells was closely associated with poor prognosis in these patients. As you can see here, the patients whose tumor had high basal expression of TG2, they showed recurrence-free survival for a very short time. And also the metastasis-free survival for a very short time. So high expression of TG2 kind of acted like a poor prognosis for these patients. This is pancreatic tumors. Tumors with high TG2, they showed shorter overall survival and low TG2 expression. Same thing for ovarian cancer, high TG2 and low TG2. So I think at this stage, we started getting convinced that it might be a very important protein or gene which is regulating something to do with multi-drug resistance. So we stepped back and started with a little cleaner system. Instead of using transformed cell lines, which already have hundreds of mutations, we started with a so-called immortal memory epithelial cells, MCF10A cells. So we introduced TG2 expression, TG2 retrovirally in these cells. So this is a parental cell line, and when it's transduced with a retrovirus TG2 gene. But we were really amazed that overexpression of this protein totally changed the morphology, and it looked like a fibroid type of tissues, which indicates the EMT phenomenon, or epithelial to mesenchymal transition. So we immediately did the gene signature for EMT in these cells, and beautifully you can see that all the epithelial-related genes, they are downregulated, and these are mesenchymal genes which are upregulated. So it clearly showed that it induces EMT. And once again, this is the same thing. But in addition, I'm just going to go very quickly. It promoted the invasiveness, the TG2 expression. It promoted the anchorage-independent growth of these cells. It promoted their motility. It promoted the stem cell-like phenotype. As you can see, these are mammosphere, parental cell line, and when you overexpress TG2, there's a very high efficiency of colony or mammosphere formation. In 3D cultures, these cells, when you culture the normal cells, they form very well-defined acinar structures. But when you overexpress TG2, they lose that polarity, and as is shown here. So these are low TG2 or TG2 deficient cells. They form beautiful single layer which survives, and these are overexpressing TG2. So they lose that polarity and they grow like tumoroid. This is a basement membrane, nicely laid out basement membrane where these cells, they attach and they survive. And they lose the basement membrane as shown in laminin 5. This is an E-cadherin expression that's totally lost in TG2 overexpressing acinar structure or tumoroid structures. So then let's see if you knock down the TG2 expression in a drug-resistant cell line. So it's a breast cancer cell line, highly resistant for metastasis, which I showed, highly resistant to chemotherapy, which I showed in the first slide. When you downregulate TG2 expression, majority of the cells, they undergo cell death by apoptosis. And this is a pancreatic cancer cell line. When you downregulate TG2 expression, you see a massive accumulation of these autophagosomes, which is a typical of process called autophagy, which was discussed yesterday in one of the lectures. So molecular mechanisms, I have so just I'll quickly run that. What it does, that TG2, when you overexpress TG2, it constitutively activates NF-kappa B. It does not need, like these are TG2 deficient cells, you need TNF in order to activate NF-kappa B, but when you overexpress TG2, NF-kappa B is constitutively activated. And we have worked out all the mechanisms. And TG2-induced NF-kappa B binds to the HIF1 promoter, and as shown here by CHIP assay, you can actually see increased or binding of TG2-induced NF-kappa B, and it's in complex with TG2. So both TG2 forms a complex with P65, sits on the promoter of HIF1 and upregulates its expression. And both NF-kappa B and HIF1, they can modulate several genes which contribute to metastasis and drug resistance. And here you can see that constitutive activation of HIF1 in TG2 overexpressing cells and no expression in deficient cells. And when you induce hypoxia, it's even superinduced. So we wanted to see if it's really that's the case, can it be a good therapeutic target for treating the drug-resistant tumors? So we started with pancreatic tumors. It's a PANC28 cell, which is highly aggressive, very high expression of TG2 in these cells. So with time, you can see increase in the tumor size. When you treat with gemcitabine, you do see some reduction in the tumor. And when you treat with liposomal-based siRNA TG2 specific, you see as good impact as is the gemcitabine. And when you combine TG2 siRNA with gemcitabine, that effect was even better in a very aggressive tumor. Here is the tumor size. And even more impressive was the dissemination of these tumors, local dissemination. It completely knocks down the dissemination of the pancreatic tumors in the local sites. Here is a ovarian cancer. Once again, HA8 cell line, which is highly invasive and metastatic. This is TG2 siRNA, and this is combined Taxol plus TG2 siRNA. This is drug-resistant HA8 cells. Once again, you see a very nice effect of TG2 inhibition by siRNA. And we can see that these liposomes, they do deliver the siRNA. siRNA is labeled here with red fluorescence. It's nicely delivered to the tumor cells. It's a large magnification. You can see the liposome sitting inside the tumor cells. So let me summarize that what we learned so far, in the tumor milieu, the immune cells, they're producing continuously the cytokines and chemokines, including like TGF-beta. Prolonged exposure of these epithelial cancer cells to these factors hypomethylate the TGM2 promoter and leads to its constitutive activation. And once TG2 is overexpressed, it results into the constitutive activation of NF-kappa B by a known canonical pathway, and NF-kappa B then activates TG2. So it's a vicious cycle, which is a positive feedback loop is initiated. NF-kappa B can activate HIF1, which I showed, and HIF1 can activate different pathways here and genes. And it can lead to the EMT and stem cell-like traits, contributing to these different so-called the hallmarks of cancer. And all these together, they lead to the metastasis and chemoresistance. With that, I would like to stop here and would like to welcome any questions or comments, either now or after the lecture. Thank you very much.